Hi everyone, I'm Lois Akosia Uklu, and then I'm presenting on my work, which was on analyzing the biodegradation potential of microbes on local dye effluent. So, in our textile industries, we have two groups being the heavy textile industries and then the small scale local textile industries, which in our setting we call them the tie and dye stations. Now, with these heavy textile industries, there is a form of treatment of their wastewater effluent prior to disposal into our sewage systems. Whereas on the other hand of the small scale industries, there is no form of treatment. Hence, the effluent is directly disposed into these drainage systems. And this could be harmful because the dye effluent contains sulfur compounds, azo dyes, heavy metals, and hydrocarbons. And these are known to be allergic and allergens and then toxic to both human and aquatic life. Hence, there is a need to employ a more safer treatment method, which is the biodegradation. The use of physical and chemical methods introduces more toxic compounds, and this makes bioremediation a more safer approach as it uses microbes like bacteria, enzyme, and fungi to break down organic molecules in dye compounds. Hence, the work focused on isolating, characterizing, and assessing the biodegradation potential of these microbes on local dye effluents. So I obtained water samples or effluent samples from Nima, which is a suburb in Accra, and also soil samples around the textile dye workstation area. A serial dilution was performed, and then the isolates were plated on various media to separate the organisms or the isolates, and they were grouped according to shapes and color and sizes, and hence a gram staining method was done to differentiate them into gram negative and gram positive bacteria. A further biochemical test was done to characterize the isolates, and then the various results obtained were compared to literature charts to know which organisms we isolated or found out. In the bioremediation process, the effluent was initially sterilized by autoclaving to get rid of existing bacteria, and a mineral salt medium was added to the effluent. The inoculum of the organisms being Pseudomonas aeruginosa, Proteus mirabilis, and Staphylococcus epidermidis were then inoculated into the effluent sample, left on a shaking incubator for a period of 21 days. Now, there are some things known as physicochemical parameters. These include pH, TSS, which is total dissolved solids, TDS being total suspended solids, BOD, and then COD. Now, these parameters were all recorded prior to the degradation process and after the 21-day degradation process. So during the 21-day process, the pH and then the OD, which is optical density, were recorded after every 48 hours. Now, in the results that were obtained, the initial values of the physicochemical parameters obtained were higher than WHO permissible limits, and with the values we obtained after the biodegradation process, we could see that for some of these organisms, there were a reduction in some parameters, and then there was an increase in the parameters like BOD and COD. Now, this could be attributed to BOD being the amount of oxygen needed by microorganisms to break down these effluent compounds. Now, the more the bacteria is undergoing metabolic activity, there is a constant release of byproducts and more oxygen. Hence, the intensity of the oxygen in the sample is increased. That's why we recorded a high level of BOD and COD. TDS and TSS are measured using a glass fiber filter. When the effluent is passed through the filter, the compounds that pass through along with the water are known as the total dissolved solids, and those that are retained in the filter are known as total suspended solids. Now, these contain the harmful molecules or compounds or hydrocarbons which are found in the dye. And then there was a high level of TSS and TDS recorded. This showed that the textile effluent was highly concentrated and contained harmful and toxic compounds. Now, there was a final flux which contained a combination of the three isolates being Pseudomonas, Proteus, and Staphylococcus. We could see that at the end of the biodegradation process, there was a color comparison done with the initial effluent obtained. And there was a significant reduction in color for the effluents treated with Proteus mirabilis and the effluents treated with a combination of all the three isolates. So this confirms that indeed these bacterial isolates, or these specific ones, have some biodegradation potential, as they are able to break down these toxic molecules into less harmful forms. So the main observations were that these textile industries, since there is no form of treatment, their effluents are highly concentrated, and this predisposes both human and aquatic life to danger. And these organisms have biodegradation potential. Now, other future work could be that we employ the use of bacteria. Maybe others could also employ the use of fungi or yeast in treating these textile effluents.